So there, you got the link that I sent you.
I just wanted to tell you that maybe in the recording,
Yes.
there was one last part where it was not class, where so maybe it was not supposed to be recorded, but just to tell you.
And in which part?
In the last part,
in the
last lecture.
Okay,
yeah,
okay.
I guess you stayed over to speak.
I stayed. There was okay, okay. No, thanks for letting me know. Yeah, yeah. Okay, so let's get started. So essentially, what we've spent the last 3 lectures on is getting a sense of numbers, length scales, time scales. And one of the take-home messages of the, let's just to recap here a bit, of the last lecture is that unlike in the way we think about many models in biological physics, the cellular environment is extremely crowded. And it's crowded by numerous organic and inorganic species. And we did some very simple back of the envelope type of calculations to show that, for example, the number of proteins in a cell is on the order of 10 to the power 6, number of water molecules 10 to the power 10, number of ions 10 to the power 7. And the implication of this is that this is all jam-packed in 1 micrometer cubed. And therefore, this sort of has an implication and an effect on the typical distances and the spacings that separate different molecules in solution. So the typical distance between 2 proteins in a cell is on the order of 100 angstroms. Okay, that's just within proteins. And then we took that a step forward, tried to understand timescales. And one of the things that we sort of tried to get an appreciation for is that if one thinks of these numbers, so 10 to the 6 proteins in the cell, and if I look now at the cell cycle, so the time scale it takes for a cell to divide into 2 cells, which is on the order of 3,000 seconds, then what this means is that there's some very, very fast processes that have to happen on very short timescales. So you have to essentially generate about 1,000 proteins per second to create 2 cells. You have to suck up on the order of 10 to the 7 water molecules per second. Okay, and what we try to use these numbers to argue is that there's a lot of directed motion that's happening in biological processes. And these directed motions are dictated by chemical potentials, electrostatic fields, external pressure forces that sort of limit the phase space, if you want to call it that, that the protein or biomolecules can explore in their particular function. If they were going to do that, then we would be waiting a long, long, long time for many processes that are relevant for biological function. Okay, so this is why there's a lot of interest and time that has been spent on understanding specific directed microscopic interactions in biological systems. And okay, so that's what we did in the last class. And so now what we're going to do is we're going to move into the last element of this discussion, which is energy. We've talked about time, length, and now we're getting into energy. And what we want to try to get a feeling and an intuition on are how thermal fluctuations at room temperature or cellular temperatures and at the length scales at which they occur, so at the nanometer length scale typically, how do those, what's special about that? What's special about the different sources of forces and energies that one can think about on those length scales? Okay. And so in order to move ourselves in that direction, let's first think about two different types of biological problems that are happening in the cell. Okay, one standard one is the protein folding problem. So I have a polymer, a protein, and it has a three-dimensional structure. I can unwind it, I can unfold it, I can stretch it, I can pull it like a rubber band, and I can do some mechanical work on the polymer. And that mechanical work will be manifested in some type of, elastic energy in the polymer. And that is happening in a thermal bath. And somehow thermal fluctuations will allow for this to happen. Okay, so this is what we want to understand somehow. How is that possible that this is allowed to happen? So this is one example. On the other side, in the example at the bottom, this is a completely different process where DNA gets replicated. So you take a single DNA strand and you make two daughter strands. And in order to do that, you need to do work, you need to do chemistry, you need to create and break different types of covalent bonds. So this means that we're dealing with, we start dealing with the electronic degrees of freedom in the system. Okay, so we want to try and understand over the course of this lecture, how is it that thermal fluctuations can drive, on one side, at this particular length scale of nanometers, how can it drive protein folding, unfolding, and on the other side, how can it drive chemical reactions? Okay, where one is looking at different energetics. Okay, so that's sort of the background illustrative examples of what we're going to try and dissect a bit. Okay, so you're all familiar with thinking about forces and energies in different situations, and maybe this might be the first time that you're beginning to think about how to relate those macroscopic energies like elasticity. Okay, so elastic forces. So you stretch a rubber band. How does the analogy of that extend to when I start thinking about stretching or bending a DNA molecule or a protein? Can one think about a Young's modulus of a DNA or a protein? Okay, and in fact, one can. We're all familiar with the role of electrostatic interactions. Electrostatics is really a critical part of some of the underlying forces in that drive biological interactions and functionality. And those forces, electrostatic forces, occur in a medium. They occur in typically water. And so we need to, and that water has ions. So one of the things that we need to get a feeling and a sense of is how our electrostatic interactions modulated and changed by the solvent. And so some of you might already have, and for sure have come across this in terms of how the dielectric medium screens interactions. And on the other side, if I have ions in solution, how do the interactions between ions change as a function of the concentration of the ions, as a function of the dielectric constant, and as a function of temperature? Okay, so that's the type of thinking that we're going to try and explore in today's lecture. And so, in order to get to that point, we need to start asking questions like the following: what is the energy required to break a covalent bond? What is the energy required to break an electrostatic interaction? That is a charge-charge interaction, a charge-dipole, a dipole-dipole interaction, a dipole-dipole screened in a solvent medium. What types of energies do we need to do these types of processes? Because they all relate to important physical processes that happen in biology. Okay, so let's go back and start remind ourselves a bit of the order of magnitude of different interactions. So the strongest interactions in the world or in physical biological systems are the covalent bonds, covalent interactions. Covalent interactions are the strongest because they involve electrons being shared between two atoms. And depending on the number of electrons that are shared between the two atoms, this affects the strength of the interaction. So order of magnitude, and so now these are because so I should have put units of EV, but this is something that one should always have at the back of your head in your favorite unit that you like to use. Covalent bonds, order of magnitude, the energy range is between 300 to 800 kilojoules per mole. That is about I don't know, 2 to 1.5 to 3 EV, roughly. That's the strength. Thermal energy is 25 milli EV, or in kilojoules per mole, it is I think 2.4 or something. Okay, so that fixes for us, KBT fixes for us how we can rationalize and understand these numbers, because it's thermal fluctuations that are going to drive all the processes that we're at equilibrium, all the processes that we are going to be discussing. Okay, so that's the strongest interaction. Let's move down the scale a bit. Hydrogen bonds. So hydrogen bonds are just, they're mostly electrostatic interactions. So the chemists like to think of them as, they think of them as hydrogen bonds because of, the reasons that I explained to you in the first lecture, because of the difference in electronegativities of two atoms that are covalently bonded, and this creates a bond dipole, and this leads to bond dipole interaction. So it's mostly electrostatics. There is some quantum mechanical electronic effects, but it's mostly an electrostatic interaction. And of course, it is much weaker than a covalent bond. And so the range of interactions of hydrogen bonding or electrostatics cover a range between 10 to 100 kilojoules per mole. Now, why is there such a big range? What do you think? wait, let me, let's see who I can, Maha.
Yes.
So what do you think causes the big range in the spread of the energies of these types of interactions, given what I just told you?
So here we are talking about interaction between two water molecules, right?
Yep. Oh,
So-
well. Specifically, so in general, yes, but this number that I'm reporting here is generally for any type of possible hydrogen bond. So let's put it this way: the interaction between two water molecules is roughly about 10 to 20 kilojoules per mole. So can you think of a situation where you would have an energetics increase to 80 kilojoules per mole? Why? What could cause that?
I think it's the distance may affect.
Okay, that's
the type of atoms that are in interaction of the closest.
Okay. Yeah. Okay. Good. So certainly the geometry, the type of atoms will of course affect it, but but more generally, so this over here in this picture, we're looking at neutral objects with a dipole. So the effective interaction is a dipole-dipole interaction. But you could also have hydrogen bonding because it comes from charge interactions.
Yes.
Okay. You can also get hydrogen bonding because it involves a charge dipole interaction. So imagine having a water molecule, a dipole, interacting with an ion. Okay, it's not a strict hydrogen bonding as per the chemist's definition, but it's got the same essential physics behind it. And so depending on whether you have a dipole, charge, the different combinations can, of course, change the spread of the energetics. Okay.
Okay, I see. Yes.
Okay, but your answer was basically suggesting that.
Yeah.
Okay, good. Okay, so those are the two. Well, there's one more interaction that I forgot, but I think I have it later on in the discussion, but we can just talk about it now. So we have covalent bonds, hydrogen bonds, which are electrostatics, and then there's van der Waals forces, van der Waals interactions. Van der Waals interactions are the weakest forces.
Sorry, Prof.
Yes.
Yeah, I just want to, like, for instance, for the example you give between interaction, for the interaction between water molecule and ion. Yeah, in this case, can we still talk about hydrogen bound?
Are you a chemist?
No.
Oh, okay, then good. So then the answer is yes.
Oh,
if
no,
you're
no, I am a chemist.
now you're okay. Then the answer is no. No. I mean, so the chemists they tend to have chemists have very particular strict rules about what they think and call hydrogen bonding. And so, okay, in the case of the water and the ion, okay, it's not a hydrogen bonding in the strict sense of the word or the definition, but at the same time, you've got an oxygen that's more electronegative attached to a hydrogen. This creates a bond dipole, and that bond dipole now, via some electrostatic interaction, interacts with the negative electron density of the chloride. So, you know, the underlying physics that causes the hydrogen bonding
It's still present.
is still present. This is the point. This is the point. Okay, and it's much more prevalent than chemists and also biologists like to think. Okay, because a lot of that language has been developed on more of cataloging and sort of definition-based criteria. But yeah, one can do much more and better than those definitions.
Okay.
Okay.
Thank you.
Yeah, you're welcome. Okay, so I was talking about van der Waals forces. These are the weakest forces in nature, in biology, but they're very, very, very important. And they are strongly coupled to the problem of hydrophobic forces. Okay, so let me clarify one thing here. All the interactions that I'm telling you here are enthalpic forces, enthalpic interactions. But and van der Waals are the weakest one of them. On the other side, there are also entropic forces. And entropic forces are, and hydrophobic forces are one of the most important entropic forces. We're going to go deeper into that later on in the course, but I just wanted to mention that since we're talking about forces. But so van der Waals, very weak, but collectively very, very can contribute significantly to in biological systems. And I just want to add one thing here where I think there's this is a very interesting open problem, and people are just beginning to think about this. Is more from the perspective of electronic structure. So in many of you have probably encountered, have done simulations of Lennard-Jones liquid, right? Lennard-Jones, either in my course or in Giovanni's course. And Lennard-Jones liquid is held together by attractive van der Waals forces that are purely pairwise interactions. But this is a huge sort of assumption, and one of the growing areas in the area of biophysics right now is that there's now an appreciation that many body van der Waals forces and long range many body van der Waals forces beyond a nanometer play a very important role in biological function. Okay. So it's really an open area of research. Okay, so we've summarized the three interactions. Now we're ready to make some next steps and try to understand how these various things change as a function of length. Okay, so quiz. Or, well, partially quiz. How does thermal energy scale with length? Julia.
Yes.
How does thermal energy scale with length? How does it change as a function of length?
I think it decreases.
Decreases. Okay, let's see what someone else thinks. Lian. Leanne?
Yeah, I'm thinking.
Okay. No, sorry. Yeah.
But can you clarify a bit?
Okay, let me. So, if I had, well, in order to clarify the question, we have to ask two questions. Firstly, what is thermal energy? Obviously, and once you write it down, is this something that changes as a function of length?
No.
No. Okay. Good. So this is a trick question. Thermal energy is constant. Okay. So I cannot. Thermal energy is set by kBT.
Yeah.
Okay. So it doesn't decrease. It doesn't increase. It's constant. So let's keep that in mind. Okay. Okay. Now, second question. So let's imagine doing.
Sorry. Hello.
Yes.
Can I ask a question?
Please go ahead.
When you mention thermal energy is not increasing or decreasing with length, I'm a little bit confused. I mean, because maybe the one I'm thinking is that maybe thermal energy could change lengths of a system, like in the old school physics, you know.
No, okay. So let me try and clarify this. So let's so thermal energy is fixed by Boltzmann's constant and temperature. We agree on this point. That's kBT.
Yes.
Okay. Now, then it's another question to ask and say, thermal fluctuations may change and induce changes in the lengths of my system or object. This is a different thing altogether.
I see. So by thermal fluctuation is like when you increase or decrease it by thermal energy, you mean it's a constant energy?
Yes. Yeah, exactly. Yeah. Is that clear?
Yes.
Okay, good. So now we want to do a simple experiment.
Sorry, Professor, I have a question.
Go ahead, Paul.
Okay, so if you say thermal energy goes as kBT, that's right?
Yep.
But we have this lambda, which changes, which depends on temperature, the de Broglie wavelength.
Well, okay. So right now we're talking about a classical thermal energy, which is defined by Boltzmann's constant times the temperature. So
we cannot, sorry.
Yeah.
We cannot define the de Broglie wavelength of a classical system.
You can, but this would be then we're talking about something else then.
Okay.
So I'm the question I'm asking is the following. So I have think of a system that exists at different length scales. Okay, so let's go back here and look and think about this a bit more. So let's look at here. So think of a system where I have a polymer in water. Okay, so the length scale of this polymer in water at room temperature set by thermal energy is 2 nanometers. Okay, that's the length of this object here. And then we ask ourselves another question, which is now let's again fix kBT. We fix thermal energy, and we're asking what is the length of the covalent bond. That's 1.5 angstrom. So where you fix the thermal energy and then you ask a system, different systems can exist at different length scales because of their different chemical and physical properties at the same temperature. But thermal energy itself is length independent. It's fixed by the temperature and Boltzmann's constant only.
Okay.
Okay.
I get your point.
Yeah.
Sure.
Okay. Any more questions before I? Okay. So now we're going to have to go back to our one of our first courses in quantum mechanics, and we need to do this for a particular reason. I want to do some type of I say Gedanken theoretical experiment, which is the following. I want to create a chemical bond, a covalent bond of different lengths in a system. So let's imagine I take 2 carbon atoms and I want to keep them trapped at 1 angstrom, at 2 angstrom, at 3 angstrom, at 10 angstroms, at 20, at 10 kilometers, okay? And I want to know what is the simplest model that can allow me to understand the energies associated with the electron or the particle that is going to be trapped in this interaction of different lengths. Okay, so that's the question. So I have 2 atoms. I'm imagining moving them from infinite distance to close range distance. And I know that what changes inside these 2 atoms is how, in the very simple, an electron reorganizes between those 2 atoms. Okay, so the simplest model that you can use for this is the particle in a box. Okay, that's the baseline thing that you all learned in your first course in quantum mechanics. Okay, so now what I'd like you to do is I want you to make a plot of thermal energy as a function of length. It's constant, and the energy of a particle in a box. So recall that the energy of the particle in a box, how does it scale as a function of length? Alex?
1.
Oh, wait.
You mean a free particle inside a box, or the potential?
Yeah.
I mean, the free particle in a box should have 0 energy inside the box and infinite outside, right? It's very hard potential.
So how does it change as I change the length?
Of the box?
Yeah, of the box.
Well, the energy per se, I don't think it should change. What changes is, I think, is in a quantum mechanics system, the gap between the energy levels. Yes.
And how did it changes?
The gap decreases with the size of the box. Yes.
Perfect. It decreases because it goes as 1/L squared.
Yeah.
Okay. Okay. Perfect. So now, what I'd like you guys to do is to start by making a plot, thermal energy. So make a log plot of energy, distance, length. Thermal energy is constant, and the energy associated with the particle in a box, the energy levels goes as h squared over 8 mass of electron times L squared. Okay, that's okay. So I'm going to also, let's try to do it together. Where are you?
So this is just two plots, right?
Yeah, on the same axis.
Okay.
Yeah, and if you do it on the log scale, it'll be log scale. It'll be much easier for you to make sense of later. Okay, let's see. Share. Okay, here we are. Okay. So let me be really stupid and this is joules. Wait, let me check. Let me check. So I need thermal, I need, yes, thermal energy. Yes. Okay. Okay, so that's constant, independent of length. Particle in the box. So I need Planck's constant. So 6.62607.015 minus 34. I need the mass of an electron. Yes, okay, 9.10938.356. Okay, I think I need to, oh, yes. Fine. Okay, so then I have h squared over 8 mass of electron L squared. And I believe I need to multiply these by 1,000 for the units. Yes, okay. Log plot. FCN1, FCN2. And this will go from this is so the length here is in meters. Okay. So this is sort of what it should look like. Okay. Now, look at this guy. So there's the energy of the particle in the box. There's thermal energy, and obviously there's an energy, there's a length scale at which you can ask the question at what length scale do these energies converge? Do these intersect? Okay, and you're already beginning to see that they intersect in this regime here that's close to nanometer. That's not by chance. Okay? So particle in a box energy and thermal energy converge, intersect around the nanometer length scale. Okay, so that's number one. So was everyone able to?
Sorry, Ali, why do you multiply by 1,000?
Because it's the units of H are in kilograms, so I'm converting into grams.
Ah, okay, okay.
Yeah.
Okay.
Diploma students, is it okay? Everything?
Yes.
That's you, Leanne, right? Sorry, I sometimes can't see. Everyone else? Can I move on to the next thing? Okay, if you.
I see some yes in the chat, so you can.
Wait, I can move. Thanks, Leanne. Okay, perfect. Okay, so good. So now let's go back to over here. Okay, so so that's just okay. This is an extremely crude, very approximate, missing a lot of things of the question: what is the energy required to bind an electron into some confined space as you change the length of that space? Okay, but the take-home message and the picture that eventually emerges is not going to change. Okay, so this is number one. So and it's a bit of a stretch, but if you're thinking about chemical reactions in biology, okay, and you want to understand how those energies change as a function of creating different chemical bond lengths, this is a reasonably good start at getting a sense of those numbers. Okay, next one. Now, I want to look at elastic energy. So I want to ask the question, I have a rod, I have a material, and what is the energy required to stretch it by some length, delta L, and how does that elastic energy scale as a function of that length? Okay, so this is what Hooke's law. And what goes into it is you need the Young's modulus of your material. So the elastic energy will well, okay, let's wait. Let's go here. Jonah.
Yeah.
How does the elastic energy change as a function of the stretching length?
Yeah, so you need the Young's modulus, then the strain. So you need sort of the area of the length.
The length, yes.
And also the stretch, that is the displacement squared. Yeah.
Perfect. Okay, excellent. So the effective scaling of the energy is the displacement squared. It's L, it grows as L squared.
Yeah.
It grows as a length squared. Okay, fantastic. So now let's go back to my thingy here. FCN3, well, for what should I use? Well, yeah, I'm going to use the typical, I think the typical order of magnitude Young's modulus is like 10 to the 8th for a typical material. But anyway, it won't change what the message of what this is going to be. So FCN3. Okay. So let's try to make this a bit. Okay. So interesting. Something interesting is going on here. Okay, so very, very, very different sources of forces and energies. On the one hand, electronic, on the other hand, elastic, are all intersecting at one length scale. This is the only length scale at which all these different forces intersect. And they intersect with thermal energy. And so at no other length, you know, go very large in length, you will not see this intersection. Go very small in length, you will not. But there's a sweet spot, and this sweet spot is why, whenever people talk about nanotechnology, nano this, nano that, the reason for that is because of this peculiarity. No, it's not really a peculiarity. It's the fact that it's only at these length scales where thermal energy intersects with these different forces, different energies of different origins. And so thermal fluctuations at this length scale can easily drive the conversion of one form of energy to another, which is really the essence of biology. It's the essence of life. Okay. And of course, it's more nuanced than what the way I'm describing it, but that's the effective picture.
May I ask you?
Yeah, please go.
What happened if how's the intersection if we sum the potential energy of the particle in the box and the Young modulus energy intersect with the thermal energy? Because I think maybe here your purpose is to find out the bond length, and this make me remind about the Bohr radius, and it's the when we balance between the energy of the kinetic energy and the potential energy. And I'm wondering what happened if here we sum the potential energy and we see the intersection with the kinetic energy, and kinetic energy here is the thermal energy. So, yeah.
Okay, so let me just try to understand your question. So you're asking if you sum the elastic energy and the particle and the box energy.
Yes.
And you're asking what happens.
Yeah. I want to see the intersection with the thermal energy when we sum them.
You want to see how the let me thing. Well, if you, so we can sort of look at it here. So if you sum these two, you're going to get something. Well, let's see, the slope of yeah. So these are, one is L squared, the other is inverse L squared. So it won't be constant, but if you sum these two, this would give you sort of a flat line, right? More or less. But I'm not sure it's so physical to do, actually. I mean, okay, I think it's a fair question to ask. What happens if you sum the?
You can just change the code.
So you want me to do it for you? Okay, fine. I will humor you. Just.
Thank you.
So let's see, FCN2 plus FCN3 and
okay.
Yeah, well, okay. It's dominated by this guy. You can see it there. I can get rid of FCN2.
I think this is even a good result, a better result, because we see the scale is like 10 to something, like 10 to 10 to power 10.
Yeah, I don't.
If we just add the potential energy of the particle in the box and the thermal energy, I think it's around 10 to minus 7 or 6, and I don't think it's a good result. Is that?
So I'm confused by what do you mean by a good result? What does a good result mean?
So I think the bond length is Angstrom. Yeah.
So the bond length argument is just yeah, this is yeah, it's a very effective model. So I mean, don't take it too literally. It's all you should take away from these plots is that around the angstrom nanometer scale, there is this confluence or convergence of these different types of interactions. Then, if you want to answer more precisely what the specifics on the interactions in the bond, then for sure you have to account for electron correlation and blah 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 blah. But that really, this is not that's a second order complication to the model.
But I think so. The way we define the ball radius is to equal the kinetic energy and the potential energy. And if we don't care about the potential and I mean the Young elasticity energy,
yes.
so they should somehow also, like, at least if it's not Angstrom, it should be like nano something. But it's 10 to minus 7 or 10 to minus 6 is a bit far from.
So okay, so your point is that you think that the model that's going into defining FCN2 is wrong, or what?
I mean when we add the contribution of the Young elasticity energy, and it's show a better result.
Okay, let's talk about this. offline. Okay.
Yeah.
Okay.
Thank you.
Okay. Thanks. Okay. So good. So let me stop share. Let me go back to here. And okay, so you can do many of these fun exercises on using the simple models and get a feeling of these. Sorry, I think someone needs to be muted. Hold on a second. Okay, thanks.
Sorry, Professor.
Yes.
Yeah, you said there is convergence at around the nanometer length.
Yep.
Yeah, but according to the picture, as if I see that one is it is happening for the particle in the box there.
Yes.
Yeah. What about the one?
Yeah.
With the
Yeah. Of course. So sure. So okay. This is so. Let's go back. Share. So the details of it depend on how you choose the Young modulus parameter and things like that, which I haven't done entirely correctly. And the point is not to say that it is exactly 1 nanometer. The point is to say that there's a range. So hold on. Where are we? Let's go here. FCN3, FCN2. So, there's this range of energies where things are in this length scale of angstrom to nanometers. It's not 1 nanometer. Okay, that's so, and the details of the slope and everything depend on how you choose the exact value of the Young's modulus. It depends on how you choose the surface area and the length and details like that. But that's not the point. Okay.
Yes.
Okay. Okay. So let's now. Okay. Let's go back to this. So this what I'm telling you today is actually very nicely described in this and much better described, I should say, by Rob Phillips in this article, Biological Frontier of Physics. So I highly recommend you to look at this. And it has this very interesting message from Paul Dirac, who's, the father of physics in many perspectives, and a giant in the field, where he makes a very interesting remark about the rules of life and the role of physics there. But I highly encourage all of you to take a look at this paper, because it besides going through these arguments, he also describes other interesting facts related to it. Okay, so this is the take-home message. Length, energy, thermal energy, and there's something interesting going on here. And so order of magnitude, the forces that thermal forces that are relevant in these types of situations are on the order of 4 or piconewtons, essentially. Okay, that's the magnitude of the forces that one should be thinking about. Okay. Questions? So I should say that, you know, I haven't done this myself, but you can ask also the question, if I have charges, let's say spherical charges, and I want to create like a protein, a spherical shaped protein, or like a virus with a particular radius. You can also ask, what is the electrostatic work required to create a sphere of a particular radius, and that grows as r cubed, and you know, basically you reach a very similar conclusion, which is that there's something interesting going on only at this length scale. Okay, at this nanometer length scale, and so there are some other interesting things now to observe in the context of why this nanometer length scale is interesting and special. And this is the length scale at which, you know, many of the molecular forces and interactions that we think about and study in biology are relevant. Okay. So now we're going to spend some time dissecting some of those forces. So the first one, and I've alluded to this, is electrostatics. So electrostatics are probably one of the most important interactions, and that's why, you know, people spend a lot of time making sure, or they spent a lot of time back in the day making sure that electrostatic contributions to energetics were taken care of correctly, and this is something that for the diploma students we already discussed in the numerical class. So I have two charges, and the electrostatic energy, of course, depends on the distance and on the effective dielectric constant. And a very useful way of understanding how these electrostatic interactions change as a function of the dielectric medium is through something known as the Burum length. So the Burum length is the length at which electrostatic energies get much larger than thermal energy. Okay, and so basically the electrostatic energy between two charges goes as E squared over the dielectric constant of the medium, and you can ask the question, at what length does the electrostatic energy, the screened electrostatic energy, equal to thermal energy? Okay, that length is known as the Burum length. And so for water, which has a static dielectric constant of 80, if you do the math, the Burum length is about 0.7 angstroms. So, you know, nanometer, let's say, sorry, 0.7 nanometers, 7 angstroms, but on the order of a nanometer. So beyond 1 nanometer, electrostatic.
Sorry, Professor.
Yes.
You said what is this D? Which constant?
This is the static dielectric constant.
And again.
So hold on, Pelush. Just so Paul, so in continuum electrostatics, you can effectively account for the dielectric medium in a mean field way using an object known as the dielectric constant. And the dielectric constant is related to the underlying dipole fluctuations in the system. Okay, so it's a continuum model or effective model that describes the dielectric medium of the solvent.
Okay.
Okay. Okay. And so just before I get to you, Pelush, so what the dielectric medium does is it screens interactions. It screens the electrostatics. So I'm sure if you go back to your first course in electrostatics, you took a parallel plate capacitor, you looked at the field, and you ask, well, how does the field change when I have a dipole field because of the dielectric, and that field decreases? So there's a screening. So the dielectric medium screens the interactions between the charges. Okay, go ahead, Pelush.
Yeah, I was just confused. I think I misheard you. At this length, did you say one of the forces is larger, or they are getting closer to each other?
So no. So what I'm okay. Let me back up a bit. So I'm asking the following question. So imagine. So let's first ask. I have two charges in the gas phase. Okay, and the charge interaction in the gas phase goes as -1/r, two opposite side charges. When I put that in a dielectric medium, it goes as -1/r epsilon. Okay, that the units of that quantity is energy. Now I can ask, at what length is that energy equal to the thermal energy? Okay, and that so that's what this equation, and because the E is fixed, the 4 is fixed, the pi is fixed, the epsilon 0 is fixed, dielectric constant is fixed. The variable is the distance, which is LB, which is the Burum length. So I'm asking, at what R, if you prefer, you can change this to an R. At what R does the screened electrostatic energy equal to thermal energy? Okay, so that was the first question. Then I said, okay, well, the length at which those two energies are the same is roughly 7 angstroms. So at distances bigger than 7 angstroms, essentially electrostatic interactions are screened by the solvent. So it's thermal energy is what drives the fluctuations and also the diffusion or the dynamics of two charges that are beyond 7 angstroms in solution. But when I get close to 1 nanometer, I get to about 0.7 nanometers, then the two interactions are equal. And then when the distance gets even shorter, then electrostatic energy gets much larger than thermal energy. Is that clear?
Yeah, it is clear now.
Okay. Okay. So, well, why is this important? Well, this 5 angstrom, one nanometer length scale is the length scale that defines many specific biological interactions. Sorry. Sorry, Tasian, there's a lot of you. I had to mute you because there's a lot of background noise coming from your microphone. Do you have a question?
Okay, let him ask from my computer.
Okay, this is Tasse from Paulo's computer.
Yes.
Oh, I see this relation to find like equilibrium length 8b violate the bond length. I think. Can't we add like the if we have at 0 temperature, we'll have a 0 8b.
So I really don't understand your question. I can't hear basically anything that you're saying. Can you please type your question in the chat and then I'll get to it.
Let me repeat again.
Okay.
I think you will understand.
No, I really can't. So
okay.
let's
type it.
Try again.
This equation to find LB.
Yes.
At 0 temperature, we can find infinite length. Is it?
Yeah. Well, okay. This is so. This we're talking about electrostatic interactions in at finite temperature in solution. So the question of 0 Kelvin is then dictated by very different physics. So then you'd be looking at quantum fluctuations and things like that. So this is not, here we're talking about classical electrostatics at room temperature in a solvent bath. Okay, so that's.
I was thinking about the bond length and
the bond length. Sorry, the bond length of what?
The bond length of the two, like a covalent bond.
Okay. So first of all, this is not. I don't think this is a good. If you want to discuss covalent bond, then we have to use a different model. This is about.
Okay.
Okay. This is about a positive and a negative charge in solution, in a dielectric medium, at room temperature under classical electrostatics.
Okay.
Okay. Okay. All right. So just to give you a feeling of how the Burum length changes for different solvents. So the number that I reported to you was for water. Okay, but these are two different solvents. I don't even remember what their names are, but it's not important. Well, maybe, well, Romina, do you know the names of these solvents?
Toluene.
Which one?
Toluene.
Which one is toluene?
The top.
The top is toluene, and this one is?
I don't know.
Okay, all right, fine. No, it's okay. Okay, so these are non-polar solvents. Okay, and you can do the same exercise, which is look at how the electrostatic interactions change as a function of the distance and at what distance they're equal to thermal energy, and you see that the Burum length the length at which you go from thermal to strong electrostatics changes drastically, depending on the type of dielectric, the dielectric constant of the solvent that you're in. And now this raises an interesting point. The point is that, well, nobody questioned me about this, but I assumed that I could use a dielectric constant of 80 to describe my interactions in the liquid. And I assumed that that dielectric constant is correct and effective for biological systems. But why? Why should that be the case? And in fact, it's not the case. In fact, if you look at, people have now studied this with both from an experimental view and also from theory, is that the effective dielectric constant of in biological settings may be very different from the 80. And now this should be not so surprising to you because of this stuff. Where are we? Right? You've got a very, very crowded environment, and that crowding can and does affect the dielectric constant. And so this is something that's still not completely understood, and what people think and suggest is that the dielectric constant in these confined systems reduces significantly. So it can go all the way from 80 right down to 10. And if that's happening, then the range of length scales over which electrostatic energies may outcompete thermal energy may not be 7 angstroms. Okay, so that's so I'm sort of, in some sense, building myself for a self-contradiction and putting my foot in my mouth. On the one hand, I'm telling you that well, this length scale is very important, and there's the Burum length in dielectric water is on the order of 7 angstroms, but the dielectric constant can vary. It may not be 80. But okay. So even if you go down to 10, even if you go down to 5, okay, you're still on the order of energies that you're looking at here. It will change things a little bit, but it's not going to be such a drastic change. Okay. Okay. Okay, I think I'm going to stop here for today, and we will continue tomorrow. So we have we'll finish tomorrow with looking at the by screening, so how ions behave near near biological systems. What are the typical ways to think about these? And then we're going to move into sort of a different topic, now focused on the role of water in and sort of reviewing why water has many special properties and why they're relevant to biological systems.
By the way, may I have the slide of the?
Yeah, yes, you. Yeah, I will. So okay, what I'll do is I will, because I'm changing them basically every day before I teach. So I will give them until today's lecture. Okay, I'll put those on Slack.
Yeah.
Okay.
Thank you.
Okay,
thanks, Leon. Okay.
So can I ask you something?
Yeah, please go ahead.
Yeah, I want you to give me the name of the I mean, the application you use it to plot the energy?
I use Mathematica.
Okay, thanks.
Yeah, but you can use many other things.
Yes.
Okay. Any other questions? If not, just to remind you, tomorrow we also have class at 9 a.m. at the same time. All right. Thanks a lot, guys. Have a good day.
Thank you. Bye-bye.
Ciao. Ciao.
Bye.